Is that too loud, or? Okay. So I'm going to talk about some of my obsessions. You've already heard. I'm a bit obsessed with open science. Not going to talk about that at all. I'm a bit obsessed with social media and science. I'm not going to talk about that really at all. Randy didn't mention this. This is a bigger obsession, more of a lifelong thing. Apologies to any non-Red Sox fans out there. Not going to talk about that. But what I am going to talk about is microbes and microbial evolution. And amazingly, I did not realize this, but I have been a bit obsessed about this for a long time. So in 9th grade English class, we got an assignment where you could write your own essay question and then answer it. First of all, ignore the handwriting. That really is mine, but I am embarrassed about it. And my essay question was, describe one step in the evolution of a bacteria. And then I answered it in 3 or 4 pages. I don't remember this at all. And I went to college to be an East Asian Studies major and eventually switched to biology. And as I was telling Jennifer earlier, one of the reasons I switched to studies of molecular evolution was I took a class as a sophomore from a graduate student, Jennifer, on molecular evolution that just changed my life and got me really interested in combining ecological and evolutionary studies with molecular biology. So that's what I'm going to talk a little bit about here, my obsession with microbes and microbial evolution. So in this context, I've done a lot of work on microbes and microbial evolution, but this day and age, we talk about this in terms of the microbiome. That's sort of the hottest word, hottest topic in microbial studies. And this is a plot of PubMed, hits to the word microbiome, and it keeps going up almost exponentially over time. There's just an incredible rise in studies of microbial communities. I'll talk about what the microbiome is in a few minutes. There's been this big effort to characterize the microbes on human. There's a European effort, an NIH effort, a variety of other efforts on the human microbiome. There are microbes and microbiomes in the news all the time, in good and bad ways, and now there's a lot of discussion of how we may either help or hurt our microbiome in a variety of ways. So it's become this incredibly hot topic. If you're interested in what it means, I've actually written a couple of blog posts and articles about this. Basically, the way I view the microbiome is the collection of microbes that live in a particular ecosystem or biome, the total content of that microbial community. Sometimes people use this specifically to refer to the genomes of those organisms. I usually use it to refer to just the collection of organisms in a system. Microbiome studies have gotten the most interest from studies of the cloud of microbes that live in and on people, but as, of course, many of you know, here, there's a cloud of microbes that live in just about every ecosystem on the planet or every place on the planet. Many of these are associated with plant and animal hosts, and those do some very interesting things. But there are also microbial communities in the soil, in the water, in the air, and every other place on the planet. And of course, Novozymes has specialized in making use of some of these microbes for a variety of systems. But there's microbes everywhere. In general, now people are using this term microbiome to refer to any community of microbes. So one question that I'm sort of really interested in is, why has this become such a hot topic now? What has gotten people so interested in microbiomes most recently? And I'm just going to, as an introduction, go through what I think are some of the explanations. One of them is this growing appreciation of microbial diversity. So there have been, over the last 30 or 40 years, an increasing number of studies of microbes in various systems shown to be important in the health and function of a particular ecosystem. Of course, this started with the discovery of microbes, but there's an incredible diversity of form that has been characterized in microbes, and a growing appreciation of this diversity of form. There's an incredible diversity of phylogeny, that is, the different types of microbes scattered across the evolutionary tree. And of most interest to people is contained within microbes is an incredible amount of functional diversity. Chemical diversity, in particular, is what a lot of people have been interested in. And of course, we microbiologists think that microbes run the planet, so we think that they're pretty important to study. Another reason that I think the microbiome has grown in interest is what I call the post-genome blues. So, as you heard, I worked at TIGR for 8 years, sequencing genomes, indirectly associated with some projects on some of the larger organisms, but mostly worked on microorganisms. And during that time, there was a lot of selling of the benefits that would come from sequencing the human genome or sequencing a plant genome or sequencing a microbial genome. And let's just say some of those benefits did not come into being immediately. So there's been lots of articles written about this. Did we oversell the benefits of the human genome? And of course, there are reasons why the genome might not explain everything. There's, of course, the gene content only explains what the potential is. Now we have to characterize the transcriptome, and the proteome, and the metabolome to understand what's actually encoded from the genome. There's epigenetic tags on organisms where the individual genome sequence is not the only thing that tells you what's going on in the organism. There's something called the variome, which is the variation within individual multicellular organisms. There are different genetic content in different parts of an organism. And the microbiome is just sort of in this context, yet another thing that it can explain basically why the genome itself does not explain everything. Another reason for the increased attention to microbiomes is technological advances. And you know about all these in terms of a variety of studies of biology, but for microbes, this has been particularly important for a variety of reasons. And one of the main reasons is that, as I assume most of the people at Novozymes know, if you go to any community in the environment like this Yellowstone hot spring and you collect a sample and you split it in two and you look at the sample in a microscope and you count the number of microorganisms that you can see there, or you take out a sample and you grow it in the laboratory and you count how many different things you can grow, what you see is that there's a much greater number of things that you can see in the microscope than things that you can grow in the laboratory in pure culture. And this is generally known as the Great Plate Count Anomaly, or a variety of other acronyms, and it's been a big problem in microbial studies. There are many ways that people have tried to deal with this, but in the 1980s, primarily, Norm Pace and colleagues developed an alternative way to study microbes and their communities by going out into the environment and isolating originally RNA from the environment and now DNA from the environment, sequencing the DNA from those environmental samples and using analysis of the DNA to indirectly characterize what microbes were present in that environmental sample. A lot of this work was based upon the work that had been done by Carl Woese, where he had taken cultured organisms and shown that you could characterize what kind of cultured organism you were looking at by sequencing ribosomal RNA genes from those organisms. So isolate ribosomal RNA originally, and now the DNA that encodes ribosomal RNA, sequence it, line up sequences from different organisms, compare them to each other in much the same way that you compare the skeletons of vertebrates, look at the similarities and differences, quantify those similarities and differences, and then build an evolutionary tree that tells you what kind of organism you're looking at compared to other organisms that have been characterized. This is what led Carl Woese to discover the third branch in the tree of life, the Archaea. And this general concept is basically what Norm Pace and colleagues took and applied to environmental samples and used it to indirectly characterize organisms by sequencing their ribosomal RNA genes and then placing them in the context of known taxa by phylogeny. That's something generally known as phylotyping, which I will come back to. What I'm sort of generally referring to as CSI microbiology can be applied to a variety of microbial communities and has. One thing that people are doing now, which I will come back to, is rather than just sequencing ribosomal RNA genes from environmental samples, we can now sequence the entire genomic content from an environmental sample, something generally known as metagenomics. Apply the same general methods to those metagenomic samples with a little bit of variation that I'll talk about in a while. You can figure out what organisms are in those samples by building evolutionary trees of sequences from those samples. You can also, very importantly, make predictions of the functions of the organisms in those samples by running the same tools that people had developed to characterize the genomes of individual isolates in the laboratory. You can predict differences between communities and try and infer what are the likely explanations for those differences as well. Just as an aside, if you've seen a bit of the press recently, there's a bunch of new papers arguing that Archaea may not be the third branch in the tree of life, that they may in fact be a subgroup. Sorry, I drew this backwards, that eukaryotes may be a subgroup of Archaea. I'm not going to really talk about that in detail, but what that has come from has been analyzing genes other than ribosomal RNA genes contained within genomes of organisms. And it doesn't always give you exactly the same picture as ribosomal RNA, although the picture is frequently very similar. So Why have microbiomes gone really crazy? That's because sequencing has gotten cheap and easy and efficient. And as the costs and difficulty of sequencing have gone down, people have applied this to all sorts of organisms. But in particular, microbes have been revolutionized because, again, we can't grow many of them in the environment, and we can use the DNA sequencing to characterize their diversity in a variety of ways. Sorry, go back one slide here. So the last reason that I think the microbiome field has gone sort of crazy has been studies of a few microbiomes in particular have shown that they carry very important functions that were probably unappreciated in the past. And in particular, these are the clouds of microbes that live in and on plants and animals. So the probably most important study from this was done in Jeff Gordon's lab by Peter Turnbaugh and a few others, where they showed that if you take microbes from a genetically defective mouse that is obese with defects in the OB gene, those mice have different microbes in their gut. Not that surprising. It wouldn't have to have anything directly to do with the obesity, but they have different metabolism. They took those microbes, transplanted them into germ-free wild-type mice, and they got fat. And so this blew people's minds, basically, and led to an enormous increase in the attention paid to microbial communities, because it wasn't just that they could differ between different health states, they could transmit health states to individuals, at least in this mouse system. So in this context is where we are in microbiome studies basically now, and what we do in my lab is try and characterize various microbial communities. And this is what I'm going to sort of go through in my talk, is the challenges and opportunities in this area. So there are many challenges associated with characterizing entire communities of microorganisms. One of this relates to the diversity of microbes. There are estimated now to be thousands of different major lineages or phyla of bacteria and archaea. We understand the functions and the diversity of maybe 10 of those. The amount of microbial diversity that is out there in terms of total evolutionary diversity is enormous. There's even diversity within species that is sort of very different than what we see within plants and animals. So 2 E. coli strains can differ by 40% of their genome content. So we have all this evolutionary diversity, and then even within species, we can have massive differences within the genetic diversity. And that leads to differences within functional diversity, both within and between species and across the tree of life. Another issue of the complexity is that the way we're characterizing communities right now is mostly go to an environment, swab a sample, collect some fecal material, collect some soil, grind it up, and run short-read DNA sequencing technology on that sample and try and infer what's going on in the community of organisms, from thousands to hundreds of thousands to now billions of sequence reads. And that's very challenging to interpret that data. And there's also, of course, as I mentioned before, lots of variation within hosts. That's both due to the microbes and due to other features like the variation in transcription, the variation in epigenetics, the variation in the environment that individuals see, the variation in the genome content, and so on. So in this context, we're trying to understand the structure and function of microbial communities, especially those that live in and on plants and animals, and it's just a very complicated issue to try and tease apart. A second aspect of this complication that I think is really important to think about, it relates to public understanding. And this is a big challenge in the area of science in general, but microbiome studies, I think, may be exacerbated in some ways related to this. On the one hand, we have lots of people, probably many in this room, that are what you could call germaphobes. That is, they think about microbes in the context of they're going to make you sick, and most of the time they think about how we can filter out, swab, sterilize, kill, destroy, or get rid of the microbes from a particular environment. You know, you have a cold, you go in and take antibiotics, no matter what the cause of it is. We put antimicrobials in our shoes, in our clothing, on our kitchen counters, in our filters, in our cars. All throughout the planet, we're basically slaughtering microbial diversity. There's antimicrobial compounds in toothpastes, in shampoos, in just about everything that we see in the cosmetic world, and it's probably a really stupid idea. But people are afraid of microbes, so that's what happens. On the other hand, we also have this kind of psychotic other side of things, where some people are promoting microbes and the microbiomes as though they're going to save the planet in every which way we lose. So every possible human ailment has now been ascribed to being caused by problems in the microbial community in the gut, even if there is absolutely no evidence for this. There are clinics throughout the country that offer fecal transplantation for schizophrenia. Fecal transplantation. It's not actually, that's probably not the worst example. But there are, the hype has gone way beyond the science in this area. There's probiotics advertised that apply to different groups. So at the local CVS drugstore, there's a whole shelf of probiotics for kids, for the elderly, for men, for women, for dogs, for cats. For every other possible group, there's probiotics. There's a book out every week now on the microbiome and how it's going to save your life. There's all sorts of weird stuff out there in the blogging world. And I now give out an Overselling the Microbiome Award on my blog, and I literally cannot keep up with the number of examples of this. So these two things conflict with each other. I think there's a happy medium in between, so we spend a lot of time in my lab trying to work on this sort of public understanding of microbes and microbiome science in the context of the complexity that I mentioned before. I actually view this now as opportunities. The complexity makes it really interesting to study. The variation within hosts makes it really interesting to study, and this sort of germophobia and microbiomania also makes it a really interesting area to be in. So what I'm going to talk about for my talk is a few examples of where I think there are opportunities in studies of microbiomes, and where we're working on this in my lab, and many other people in the community are working on similar things to try and deal with this complexity, to deal with the germophobia, to deal with the microbiomania, and to try and raise the field up to a better understanding of microbes, both from a scientific point of view and from a public point of view. So I'm going to first sort of start easy with one of the first things that people do when they use DNA to characterize the microbial community, which is called phylotyping or phylogenetic typing. And what you basically do is you go to a community, you isolate DNA from a community, you run ribosomal RNA PCR using primers that can amplify virtually any ribosomal RNA gene from that environmental sample. You sequence the ribosomal RNA genes, and again, you line them up, you compare them to ribosomal RNAs from other organisms, and you can place an unknown organism into a phylogenetic context by where it sits on the tree, and then make some predictions about that organism, or at least track that organism in different environments by where it sits in the tree. I actually first started working on this as an undergraduate at Harvard in Colleen Kavanaugh's lab, where I spent a year and a half, including after I graduated, sequencing one ribosomal RNA gene, 1,500 base pairs. I got a paper out of this. And we characterized an uncultured bacterial symbiote that lived inside some clams isolated from coastal sediments in Woods Hole, built evolutionary trees, and predict the biology of these organisms. Now, the great thing about ribosomal RNA PCR is you can go to an environmental sample with more than one organism, get out more than one sequence, and infer what multiple organisms were in the sample. So let's say there are two organisms, you get two different ribosomal RNAs out of the sample, two different ribosomal RNAs in the alignment, and you can show that they show up in two different areas of the tree. You can count the relative abundance of these organisms by the relative number of sequences that you get out of the sample, and therefore infer something about the ecology of the sample, not just who is present in the sample. You can do this with 4 organisms, with 100, with 1,000, with 10,000, and now with cheap Illumina sequencing, you can do this with communities with hundreds of thousands of different taxons, and continue to use this counting to infer the relative abundance of all the different organisms in the sample. And so we use this method still in my lab to characterize the ribosomal RNA, just to get a lot more sequences than the one I got in a year and a half of work. But we characterize communities. You can do a variety of clustering and statistical analyses to compare the entire composition of different communities and see if, for example, communities that are hypersaline have different organisms than communities that are from human microbiome, than communities that are from soil, than communities that are from other environments. And again, because sequencing has gotten cheap, we can do a lot of this. So I've actually spent a lot of time in my lab, as have many other people in the community, thinking, okay, we're drowning in sequence data. We can't look. So I actually spent a year and a half. I actually hand-aligned the ribosomal RNA sequence from Colleen Kavanaugh's lab, built a ribosomal RNA secondary structure based upon the E. coli secondary structure, all this by hand. Took a year and a half. We can't, of course, do that anymore. We can't even look really at the data anymore. We have to build automated sequence analysis pipelines to deal with this data. So that's what we've been doing in my lab now for about 10 years, building a variety of tools to analyze ribosomal RNA sequence data in an automated manner. I'm not going to talk in a lot of detail about these tools, but basically, every 6 months we realize we're 3 years behind the data and try and come up with a new tool to deal with the massive amounts of data that are coming out. We never keep up, but we're trying. What I do in my lab is apply sort of an ecological and evolutionary approach to analyzing this data, which is a little bit different than what many people do in the community. So I believe that the history of organisms is really important for understanding their biology and understanding their ecology. So we always use phylogeny as a part of interpreting these sequence data sets. Now, ribosomal RNA turns out to not be perfect. Many people have known this for a long time. I'll just give you one example of how it's not perfect. The copy number of ribosomal RNA genes varies enormously between taxa. If you want to count ribosomal RNA sequences and think you are counting the number of cells in a community, but the number of copies of ribosomal RNA varies between taxa, it doesn't actually give you a very good estimate of the relative abundance of organisms. So we've developed, in collaboration with Steve Kemble and Jessica Green, a method to use the evolutionary history of organisms and the momentum of copy number of ribosomal RNAs that is conserved within particular branches to correct for the copy number variation and get better estimates of relative abundance of organisms in a system. Now, you can do all sorts of things to try and improve your ability to analyze ribosomal RNA data, but what we and many other people are starting to do as sequencing has gotten cheaper and cheaper is to just skip ribosomal RNA analysis. Go to communities, grind up the entire sample, skip the PCR, and shotgun sequence everything from the sample. And try and use analysis of that metagenomic data to get a better handle on the phylogenetic diversity and the functional diversity from that community. So one thing you can do when you do shotgun sequencing from a community is you can analyze genes other than ribosomal RNA and use them to infer information about relative abundance, phylogeny, and taxonomy from the community. Many genes in bacteria do not vary in copy number to the extent that ribosomal RNA does. Many genes actually have more variation among close relatives than ribosomal RNA does. So there are many genes that are in principle better for analyzing communities of organisms than ribosomal RNA. The reason everyone focused on ribosomal RNA was basically because primers allowed you to focus your sequencing and amplify ribosomal RNA genes from a community, but with shotgun sequencing, we get everything. So we can now analyze the phylogenetic and ecological diversity of communities using a variety of other phylogenetic marker genes. So we spent a lot of time in my lab developing high-throughput automated methods to analyze ecology of communities based upon protein coding genes, especially single-copy universal protein coding genes. And you can assign organisms to their taxonomic group, build phylogenetic trees of organisms, use the same methods you would use for ribosomal RNA, except now with a bit better resolution and a better estimate of relative abundance from a community. You can also scan through the data and find very strange things that we still can't explain. So one challenge with ribosomal RNA PCR is that the primers don't amplify everything, and not all organisms, well, not all entities have ribosomal RNA genes. So viruses don't have ribosomal RNA genes. If you want to scan through a sample and get a handle on the total diversity of the sample, it might be better to look at other genes other than ribosomal RNA. The latest from my lab is a program, oh, by the way, everything we do is released open source, published almost entirely in open access journals. We developed a program with Aaron Darling, who was a postdoc in my lab and is now in Australia, called PhyloSift, that is a Bayesian phylogenetic approach to analyzing metagenomic data. Most of the previous methods had used phylogenetic inference methods that were kind of a cheat, methods that were really fast but weren't the best optimal phylogenetic tree inference methods. This is a new, relatively fast method to use, sort of what the phylogenetic community would say is a decent phylogenetic tree method for analyzing communities of organisms. And you can then take these methods and do the same thing you would do with ribosomal RNA, but now with protein coding genes, and compare the diversity of communities and the ecology of communities with a better resolution than you get with ribosomal RNA. A second aspect of analyzing the diversity of communities that is really important for understanding microbes is to take the sequence data and try and predict the functions that are present in those communities. I spent many years working on methods, this is where the phylogenomics term Randy was talking about, to do this for cultured organisms when we have the complete genome of an organism. Now we're trying to apply this to environmental communities of organisms, and I'm just going to show you a tiny bit of this. So basically what I did as a graduate student was develop a method that said if you have an unknown gene from a sample, from an organism, and you want to understand what its function is, what many people were doing at the time, and even today, are taking that sequence and doing a database search with a method like BLAST, where you identify what the most similar sequence is to your sequence of interest, and then you look up what the putative function is of that top match, and you predict the function of your uncharacterized gene from that top match. That turns out to be not the ideal method to use. It's much more useful to build an evolutionary tree of a sequence and all of its homologs, overlay onto the evolutionary tree experimentally determined functions, and predict the function of an uncharacterized gene by where it sits in an evolutionary tree relative to other genes that have been characterized. I showed that this could work with all sorts of different protein families. We developed automated methods to do this, not very good. Other people developed better automated methods to do this. If this sounds familiar, this is phylotyping. I basically copied ribosomal RNA phylotyping and applied it to protein sequence diversity. So just like you can predict the function of an uncharacterized organism by where it sits in an evolutionary tree, you can predict the function of an uncharacterized gene by where it sits in an evolutionary tree. And you can apply this to environmental communities. We showed this with the original Venter Sargasso Sea study with Proteorhodopsin genes, which are of incredible interest. But any type of gene that you find in an environmental sample, you should be building evolutionary trees of that gene in the homologs if you want to make predictions of the function of that gene. In collaboration with Tom Sharpton and Katie Pollard, we now have a high-throughput automated method for doing this from metagenomic data called ShopMap. The paper is actually in review right now, but the software is available on GitHub if anybody's interested in it. And just as a quick aside, there's something that we've done a lot in genomes that we're still struggling to figure out how to do in metagenomes that I thought I would mention. So if you take an organism and sequence its genome, you've grown it in the lab, and you want to predict the functions present in the genome, there are lots of tools. Of course, I would argue that the phylogenetic tools are great. But imagine this scenario. You have a gene in the genome. It has homologs in a database, and none of those homologs have been experimentally characterized. It doesn't matter what method you use to select for the genes, the top hit or the evolutionary tree or rolling dice or whatever, you're not going to be able to predict the functions of the genes using that type of approach. And so we ran into a scenario sort of like this with the study of this weird thermophile called Proteorhodopsin hydrogenoformans, produces hydrogen gas as a byproduct of its metabolism and lives on carbon monoxide. And when we sequenced the genome, if you scan through the annotation of the genome, we found homologs of sporulation genes in the genome. It's related to sporulating low GC gram-positive species, so this seemed plausible, although we had tried to get it to sporulate. Our collaborator Frank Robb eventually did get it to form these endospores. That's not what I'm here to tell you about. What I'm here to tell you about is this strange thing that we found, which turns out to be really useful, which is we took the genes in the genome, and we found homologs of these sporulation genes, but we also found, of course, hundreds of genes that had no predictable function. We called them conserved hypothetical genes. So we took the genome and ran this analysis called phylogenetic profiling, originally developed by Pellegrini and colleagues at UCLA, where you take genes and you group them by their distribution pattern across species. So you look for genes that have shared presence and absence across species. They may have no sequence similarity whatsoever to each other. And we found a list of genes in Proteorhodopsin hydrogenoformans that had shared presence and absence across species and were present in sporulating species and not non-sporulating species. And what's really interesting is on this list, many of the genes we can annotate because they had homologs in characterized organisms like Bacillus subtilis. But half of the genes in this cluster had conserved hypothetical annotation, no known function. And what we did is we went to Rich Lozic, who was the world expert on characterizing sporulation genes in Bacillus subtilis at the time, sent him our list, and he basically went through our list and then made knockouts in Bacillus subtilis and showed that every single gene on this list that was annotated as a conserved hypothetical protein was involved in sporulation. This is an incredible tool for discovering the function of genes that don't have any sequence similarity to genes of known function. And we use this all the time in characterizing culture of organisms, but we have no good methods still to use this for metagenomic communities. There are people who have tried this. We think that you can cluster gene presence and absence by community or by environmental variable like pH or salinity and come up with these types of predictions, but as far as I know, there are no sort of good methods to do this from environmental communities. So we go through this community data, we try and identify the organisms by this phylotyping or variety of phylogenetic approaches. We try and compare the diversity of communities. We then try and predict the functions present in the community, maybe with phylogenetic methods or some other approach. What we really want to do is link the phylogeny to the function. We want to know which functions are present in which organisms. When we go to a community and we grind it up and then try and infer what's going on in the community is not a bag of genes. It is a compartment of organisms that interact with each other genetically and by communication systems, and they're contained within these, cells, compartments and things. So what we really want to do is go to these communities and better link the functions that we infer in the community with the taxa and the phylogeny of the organisms from those communities. And this has been a big struggle in metagenomic studies. I'm just going to give you a couple of examples of how sort of the latest methods that we're trying to go about doing this. One of them was developed by a graduate student in my lab, Chris Patel, who had been working in a plant genomics lab where they were using this method that people have used to look at chromatin structure within eukaryotic cells, where you can cross-link DNA within a cell and thereby detect different regions of the genome that are physically close within the cell, but distant within the linear sequence of the DNA by cross-linking the DNA and then sequencing the flanks around the crosslinks. And he took this method and applied it to a metagenomic community and showed that you could use it to build basically 1 million base pair linkage maps within an environmental community to link structure and function and phylogeny in particular within a community. So again, we're getting this mostly tiny short read Illumina data, but now we can build scaffolds from this Illumina data of which genes go with which other genes and make better inferences about the community by doing this. Another approach to doing this, which people are probably more familiar with, many people have worked on. There's a graduate student who was in my lab, Lizzie Wilbanks, who's now a postdoc at Caltech, who was characterizing these pink berry communities, and basically what she used was these relatively new or relatively improving methods to sequence long reads from communities. So we used this molecular method, which is developed by a company that was then bought by Illumina, which basically gets you synthetic long reads from a community by subcloning large DNA pieces and having a barcode inserted into the same different parts of the subclones of a single DNA fragment. And then we did random shotgun sequencing from these communities and also did Pacific Biosciences sequencing. And what she was able to show was that by having these long reads, what had been an incredibly messy analysis of the environmental community from the shotgun sequence data suddenly became a little more transparent in terms of interpreting the biology of the community. And she was able to show, for example, that there were these Proteorhodopsin genes, which are these light-mediated proton pumps, that appear to be linked to sulfur metabolism in one of these organisms, which had not been characterized previously. And this only was possible by linkage with these large insert DNA libraries from the environmental sample. I'm not going to tell you about this other method that she used, but she's now at Caltech in Vicki Orphan's lab, because Vicki Orphan is one of the people who developed this method called NanoSims, which allows you to scan through a sample with a rastering ion beam, basically, vaporize nanometer scale portions of your sample, feed that into a mass spec, and look at the products that come off on a nanometer scale resolution from your environmental sample. And then you can compare different parts of your sample with each other and get information about what metabolites or what elements are in those different parts of the sample. And that's how she was able to show which organisms in this community were doing sulfur metabolism or nitrogen metabolism by mapping with this nanosims method. Another approach to sort of what I would call better methods is to get better reference data for communities. So we're shotgun sequencing these communities. A lot of what we do is compare to what we call reference data, reference genomes or reference protein families, and use those comparisons and knowledge about the thing we're comparing to make sense of this fragmentary data from a community. So for example, we need more phylogenetic markers. In our original analysis of phylogenetic markers, we found about 30 genes from across bacterial species that served as good phylogenetic markers. We've been scanning through every major lineage of bacteria, coming up with lists of phylogenetic marker genes that can tell you a lot about the diversity of those particular lineages. So for example, if you're interested in cyanobacteria in the community, we have about 600 genes from throughout the genome of cyanobacteria that can serve as a good marker for telling you what kind of organism you see in the sample. Reference genomes are really important for doing this work. When I was at TIGR, I noticed, as did a few other people, that most of the genomes that were becoming available from bacteria were from just 3 phyla of bacteria, the low GC gram-positives, also known as the firmicutes, the proteobacteria, and the high GC gram-positives, also known as the actinobacteria. So we started the first project to try and correct this, funded by the NSF Tree of Life program, where at the time it seemed very dramatic. We were going to sequence 8 genomes, one representative of each of 8 phyla of bacteria for which they were cultured species and no genomes. When I moved to Davis in 2005 and got this adjunct appointment at the DOE Joint Genome Institute, we started what we call the Genomic Encyclopedia of Bacteria and Archaea, where we've been marching our way through the diversity of bacteria and archaea, and filling in the tree, getting genome sequences from any branch that has a cultured representative but no genome sequence, starting at the base of the tree and going out so we get the most phylogenetic diversity. We did a lot of analyses that I'm happy to talk to people about later that showed that this was a very beneficial approach for improving your ability to annotate genomes, to annotate metagenomes, to discover new functional diversity. Filling in the diversity of genomes from across the tree of life is a very powerful tool. And I'm just going to give you one example of this. There was a study that got a lot of press recently of these hunter-gatherer microbiome studies where people sequenced the microbial community from the guts of some people who have been relatively isolated from the rest of the world. They did shotgun sequencing and then tried to compare the genome, the organisms that were in these samples with organisms that were, say, from westernized populations. And there have been a couple of studies like this, and one of them they found a really interesting thing, which was this, I don't know how well you can see it, but there's this lineage that is all the sequences here are present from these hunter-gatherers, but not from any westernized population. And they're related to a species called Treponema succinifasciens, and then another species, Treponema, I can't even read, nor do I remember what it's called, 2 species of spirochetes that are sequences from this community are enriched for spirochetes relative to westernized populations. Now, it turns out, despite millions of dollars spent on sequencing reference genomes for the Human Microbiome Project, that is, organisms that were isolated from humans, that we had cultures of, that they then sequenced the genomes of. The 2 organisms that they were able to interpret their data with were from my Genomic Encyclopedia project. And that's because, of course, the Human Microbiome Project got samples from westernized populations. There were none of these in those samples. There were no cultured representatives. We sequenced these Treponema genomes because they were just phylogenetically novel. But that allowed the interpretation of this metagenomic data from these samples because you had a reference that was relatively closely related to these organisms in these samples. So we basically managed to convince many people that this phylogenetic approach to sequencing was useful. There's been a cyanobacterial project run at the JGI. We had a project at Davis on halophilic archaea. There have been many other such projects. It turns out these are, I think they're great. I wouldn't say they're useless because they're very useful. But if you actually look at the diversity of life, and you look at all the ribosomal RNA sequences that we have from both cultured organisms and uncultured organisms, almost all of the evolutionary diversity of life is from uncultured organisms. So we're trying to characterize the genomic diversity of life by marching our way through the cultured diversity of organisms. It's like 1/10 of 0.5% of the total diversity of life. And so there's a new effort at the Joint Genome Institute and now at a variety of other groups to go through uncultured lineages and sequence genomes from those branches and get reference genomes and functional information and other data from those lineages to much more broadly characterize the diversity of life. One of the projects was called the Dark Matter Project. Apologize to the physicists out here. Run by Tanya Wojcicki at the JGI. We have a new collaboration with Ramunas Stepanovskis at the Bigelow Labs to do the same thing. There are many approaches whereby you can get genomes from uncultured organisms. What we're doing here is single-cell genomics, flow filtering cells, running whole genome amplification, and then sequencing the genomes. Jill Banfield at Berkeley has shown an alternative approach that works really well, which is she's and her group has gotten incredibly good at actually assembling genomes from random shotgun sequencing from samples. And assembling, they just had a paper with 200 new genomes from phylogenetically novel lineages assembled by from random shotgun sequence data. But we need many of these projects, the total diversity of life that we understand now, we probably need about 500,000 genomes to characterize half of the currently known phylogenetic diversity of bacteria. And every week, that currently known phylogenetic diversity of bacteria goes up. So I don't know where this is going yet. We don't know where it's going to end. There's a lot of diversity out there, so we need projects like this to really fill in the diversity. We need the same approach to understanding protein family diversity. Go through all the genomes, sort them, build them into protein families, make reference data like alignments and hidden Markov models and trees and motifs for those, to then predict functions from communities using any type of approach. Again, I would approve of the phylogenetic approach, but we need to do that. I haven't really talked about particular systems much here. About half of the work in my lab is on model systems and characterizing the microbes in those model systems to try and understand the structure and function of those microbial communities. We started with very simple systems, so single symbionts living inside hosts. We've actually been sequencing the genomes of those chemosynthetic symbionts with my undergraduate advisor, Colleen Kavanaugh. We graduated in a collaboration with Nancy Moran to 2 symbionts living inside a host. It turned out to be incredibly hard to use random shotgun sequencing to interpret the community where there were just 3 players in the community, host and 2 symbionts. It's really hard to characterize a human-associated gut sample with thousands of species, but we're building up to that. And so we've been working on what I would call simpler systems where we have good genetic tools in the host or good ways to manipulate the host communities on the Drosophila microbiome, on the rice microbiome, on the corn microbiome, and basically using the power of genetics of these organisms, the power of all the tools that are available of these organisms, and then treating the microbiome as a trait, just like you would treat yield or wing shape or leaf morphology, et cetera, and trying to understand the interaction between the microbial communities and the hosts by leveraging all that we understand about the hosts. And that in turn allows us to test our methods. Because if we can't get the methods to work in these situations where, for example, we have rice plants growing in the exact same soil, the exact same genotype, the exact same time point sample, how are we going to characterize human population variation? And then another area that I think is really important, which we are just starting to get at in my laboratory, and a variety of other people are sort of doing similar things, is to think about the entire system. So if we think about the people in this room, I can go and collect a sample from the people in the room, characterize the microbial community in a variety of ways, and still bang my head against the wall. Because that isn't characterizing where they got their microbes from. We get our microbes from the air, from our water, from the food we eat, from our pets, from our buildings, from our mother during birth, from our family during growth. Understanding that entire ecosystem is critical for understanding the impact of microbes on particular hosts. So we need to really understand the inputs and outputs and the entire community if we think we're going to understand the ecosystem of microbes that live in and on particular organisms. One last thing I want to mention in terms of understanding these communities and the entire system is, so what I just mentioned about people is short-term interactions with people. There's also the long-term interactions. So understanding the evolutionary history and the co-evolution of hosts and their communities is really critical. That example I showed before of the treponema, the spirochete, and the hunter-gatherers is one approach that people are using. We're basically trying to do the same type of approach. Look at the evolutionary diversity of a group of organisms, look at their microbiomes, use evolutionary reconstruction methods to infer what the ancestral character states were for the microbiomes, and then infer how the microbiomes have changed over time in these different lineages. And in turn, try and associate those changes with environmental changes or genetic changes or other types of changes that occur in those organisms. We're doing this, for example, I have a grad student working on cichlids, a great model system for diversification of organisms. That seems to be one of the best for looking at host micro coevolution. And then I want to end by talking about this other side of things, which I haven't talked about a lot, which is the public understanding. So when we want to study the science of all these communities, and I would argue it's very complicated, there's lots of reasons why we don't engage the public. One of them is this germophobia. Why are we putting antimicrobial compounds in every single thing on the planet? That seems to be a bad idea. Why are probiotics selling even though there's virtually no evidence that most of them work? And so on. So we've spent a lot of time trying to work on what I would call outreach and education, both first to other fields. So microbial diversity, we have a project funded by the Sloan Foundation to work with building scientists to try and improve the understanding of the microbial ecology of the built environment. And to do that, we have to engage engineers and architects and building scientists and others, because they're the people actually doing the work in these communities that we want to layer a microbial study onto. We do a variety of other things, as you've heard. I spend a lot of time blogging. Postdoc in my lab built something called the Microbiome game. You can download this for free, of course, because I'm delusional. You can please play it. It's actually quite entertaining. And then the last thing I want to end with is this new area, which I think is critically important for public understanding and outreach, which is what many people call citizen science. You could also call this participatory science. And this is going beyond the practitioners. So, building scientists is one thing. They're thinking about the design of buildings and, adding a microbial layer onto some of their work is, foreign language maybe to some of them, but is, probably can get them to do. What we really want to do is engage the broader public with an understanding of microbes. And the way we've been doing this is this participatory science. We ran what was the first meeting on citizen microbiology here at UC Davis a few years ago, and as part of that meeting, we invited all the people that we could find doing citizen microbiology projects around the country, around the world. Basically, there were maybe 10 of those at the time. There are now probably hundreds of citizen microbiology projects that are going on across the world. Here are some examples. So I'm on the, full disclosure, I'm on the scientific advisory board of Microbiome, which is one of these places that will allow you to send in a poop sample or a tooth scraping and get data on your microbiome back. There's a comparable project called the American Gut. There are probably 10 of these home human microbiome projects. There's an incredible project run by Rob Dunn, Noah Fear, Holly Meninger, and others called the Wildlife of Your Home Project. They just got a paper that came out a couple of days ago that got a lot of press, where they sampled the microbes in thousands of homes across the United States, having people go and swab 4 sites within their homes, and then they sequence these communities. I did the swabbing with my daughter. I still haven't looked up what's in my house. I'm kind of scared of that. And what we've been doing is sort of comparable efforts. We have a seagrass microbiome project where we're trying to engage the public and other researchers in sampling. We've been doing phone and shoe sampling at a variety of conferences to get people sort of thinking about the microbes in their world. We have a thing called Project Mercury, which is a collaboration with a group called Science Cheerleaders, which we've gone to hundreds of major sporting events and other events and sampled microbes at their events, and then sequenced some of them. We also cultured microbes from these events and sent a collection of these microbes up to the space station for a growth competition experiment. And the last one I want to mention is our newest citizen science project called the Kitty Microbiome, run by Holly Gans in my lab, where just like Ubiome, if you're interested in your cats or your cat's health, you can sign up to participate in a home kitty litter sampling project and get some data on the microbiome of your cats and participate in a better understanding of cats and their microbiomes. Yeah, it's gross. And so I think to just end here, I'm really excited about the area of microbial study, microbial diversity, and microbiomes. I'm really concerned with the complexity of the systems, with the misinterpretation and overinterpretation of what's going on in the systems. You know, the low-hanging fruit is the thousands of examples of confusing causation and correlation, but there are many other examples out there. And I think that by engaging the scientific community, by engaging these other communities, and by engaging the public, we can get to some point where people view microbial diversity as something that is interesting, something that is potentially important, but something that we need to study. And I think I will